We're going to review the synthesis of purines and the metabolism of these molecules. We have to worry about de novo synthesis, salvage of existing purine bases, and breakdown. The ribose part of purines comes from this molecule. This is PRPP. This is the activated form of ribose. It's used for the incorporation of ribose into all nucleotides, both purines and pyrimidines. The first step in purine synthesis involves this reaction where a nitrogen and an amide group is taken from glutamine and added to PRPP to make this compound. Azathioprine, an important immunosuppressive agent used in both transplant patients and as an immunomodulator in other patients, is an important inhibitor of this reaction. The synthesis of purines occurs on the ribose, and this is distinct from pyrimidines where the base is synthesized separately and then mated with the ribose. We won't go through the full pathway, but it involves adding a few atoms at a time, first from glutamine, then from glycine, then from formal tetrahydrofolate, and so on, so that the rings are built up slowly in a stepwise fashion. And then finally, in the last step, the first purine, the first bona fide purine, which is IMP, inosine monophosphate, is synthesized. This requires 2 donors from the 1 carbon pool, 2 molecules of N10 formal tetrahydrofolate. IMP, which is that purine we saw in the previous slide, can be converted to either GMP or AMP. We won't talk about the pathways in detail, but this molecule, which is not obviously IMP, which is not found in RNA or DNA, can be converted to GMP and AMP, and then these can be made into the triphosphates. The regulation of this process is extremely complicated. PRPP is a feed-forward activator. There are a number of feedback inhibitors, including IMP, GMP, and AMP, all can inhibit the first step, so that high levels of cellular purines will block new synthesis. There's also regulation of the conversion of IMP to either GMP or AMP. AMP feeds back and inhibits its own synthesis. GMP feeds back and inhibits its own synthesis. And in addition, not shown on this slide, the synthesis of AMP requires GTP, and the synthesis of GMP requires ATP. This is important because having purines requires having a balance between the 2 nucleotides. Of course, RNA and DNA have all 4 nucleotides in them, so it's not good enough just to have one. You have to have all 4, and they have to be present in the proper balance. There's another way to make purine nucleotides, and that's salvage. So existing bases can be incorporated into nucleotides, and there are 2 enzymes that do it. Adenine can combine with PRPP, the enzyme is APRT, and that can synthesize AMP. And either guanine can combine with PRPP to make GMP, or hypoxanthine can combine with the ribose donor to make IMP. These 2 reactions on the left are carried out by the enzyme HGPRT. So one reaction, one enzyme for these 2, and a separate enzyme for adenine. Obviously, these reactions contribute to the pool of purine nucleotides, and so these salvage reactions will have the ability to inhibit new synthesis by the feedback that I showed you on this slide. Notice also that this slide shows the conversion of IMP to either GMP or AMP, and so the salvage reactions have the ability to inhibit new synthesis because these are the reactions of de novo synthesis. So again, all of this complicated feedback helps to balance the pool of purine nucleotides. Purine catabolism is equally complicated. The existing monophosphates, AMP, IMP, XMP, and GMP, can first lose their phosphates. They can then begin to be broken down by the loss of the ribose to give the free bases. And the free bases are funneled into hypoxanthine and xanthine. And the enzyme xanthine oxidase has the ability to convert hypoxanthine first to xanthine and then to uric acid. And of course, uric acid is the urinary excretion product. It's important to note that a deficiency in an enzyme up here, adenosine deaminase, that converts adenosine to inosine, results in a condition called severe combined immunodeficiency, in which the cells of the immune system fail to develop properly. The reason for this is still not entirely clear, but it seems that high levels of these nucleotides will inhibit de novo synthesis, will inhibit salvage pathways, and will result in improper balance between the various nucleotide pools, and so it will result in inability of those cells to develop properly because the division of those cells obviously requires nucleotide synthesis.